Welcome back to my channel, and today in this video we are going to study about a second type of physical chromosome mapping method, that is somatic cell hybridization. In the last video we learned about deletion mapping, which is also a type of physical chromosome mapping. In this video we'll study about somatic cell hybridization, which is also a type of physical chromosome mapping. Now, what do we mean by somatic cell hybridization? So as the name suggests, hybridization means it's a fusion of two cells. Now what kind of cells? The somatic cells. So when fusion of somatic cell happens, we call it as a somatic cell hybridization. With respect to cell line, if we talk, then we have two types of cell line. One is the normal cells. Normal cells means having controlled growth, and another is called as tumorous cell, having continuous growth. So these are the two types of cell line we know with respect to their growth. When we take one cells from two such somatic cells, we can fuse both the cells with the help of polyethylene glycol. So now both the cells are fused and now it is a single cell. But the single cell consists of two nucleus. So still the cell fusion happened, but the nucleus fusion has not taken place. Now further, in due course of time, the nucleus will also combine, and now this we call it as a hybrid cells with one cell and one nucleus. But this nucleus consists of chromosomes from both the somatic cells. Now, how we use the method? The question is how we use this method of phenomenon to map any gene with a chromosome. So for this, what we do is we fuse two types of cell. One, human fibroblast, and second is mice tumor cell. We fuse both the cell with polyethylene glycol. Now after fusion, this is one cell, but still they have two separate nucleus. We call this state as a heterokaryon. Karyon means nucleus. So still they have two nucleus, two types of nucleus, hence the word hetero. This stage is called heterokaryon. Now eventually in due course of time, the nucleus will also combine, and now we call the cell as a hybrid cell. But the hybrid cell still consists of chromosomes from both the cell. That means chromosome fusion has not taken place. Now again, when this particular hybrid cell will start its division, cell division, in due course of time, we will see that human chromosomes are randomly getting lost from the hybrid cell. Now the question is why only human chromosomes are getting lost and not the mice tumor chromosome. Now this reason is still unknown why when two somatic cells are fused, the chromosome from one species only get lost during cell division. This is still unknown. But we know that when human fibroblasts and mice tumor cells are fused, the chromosome which is lost is from the human chromosome. Clear? So we will get different cell line A, B, C and D with different human chromosomes. Maybe, for example, in cell line A, chromosome number 1 and 2 are lost. In B, 3 and 4 are lost. Next, 5 and 6 are lost, and so on. So we will get different cell lines where different human chromosomes are lost in different cell lines. Clear? So whenever a hybrid cell is formed using human fibroblast and mice tumor cell, the reason is still not known, but the chromosome from one of the species are lost preferentially, and in this case, the loss is of human chromosome. Now, we fuse both the cells, and before fusion, what we are doing is, these are the two enzymes. Okay, that is thymidine kinase. So as the name suggests, thymidine, that means it is needed for DNA synthesis. This enzyme, HPRT, that is hypoxanthine, guanine, phosphoribosyl transferase. So from the name itself, we know that both the enzymes are needed for the synthesis of DNA. So before fusion, what we are doing is from the human fibroblast, we switched off or we removed the gene for HPRT. Whereas in case of tumor cell, mouse tumor cell, what we did is we removed the gene for thymidine kinase. Now when we grow these cells in a hat medium, so we have three lines now. Before fusion, the human fibroblast and mouse tumor cell, and after fusion, the hybrid cell. When we grow such cells in a hat medium, what we observe is the human fibroblast cell will die. Why? Because it does not contain the gene for HPRT enzyme, which is needed for DNA synthesis, and without the synthesis of DNA, the cell will die. Similarly, the case with this mouse tumor cell. Here, the gene for thymidine kinase is not present. So without the presence of this particular enzyme also, DNA synthesis won't take place and the cell will die. The only cell which will survive is the hybrid cell. Why? Because it will complement the presence of particular enzyme or gene. So TK plus from human cell and HPRT plus from tumor cell. So it will complement the presence of both the enzymes and hence only the hybrid cell will survive in the HAT medium. Now what is HAT medium? HAT means hypoxanthine, aminopterin and thymidine. So in the presence of HAT medium, a cell must possess two enzymes to synthesize DNA. One is thymidine kinase, another is HPRT, that is hypoxanthine guanine phosphoribosyl transferase. So cell lacking any of these will not able to synthesize the DNA and hence will not grow on the HAT medium. Now how we will use this phenomenon to map any gene? Now one question is given to you, that is a panel of cell line was created from mouse and human somatic cell fusion. Each line was examined for the presence of human chromosome and for the presence of human haptoglobin. The following results were obtained. Now what is the question is, 4 cell lines are given and the presence and the absence of human haptoglobin gene is also given, gene or protein. Now these are the human chromosomes. We have to map on which chromosome the gene for this protein is present. What we have to map? On which chromosome the gene for this protein is present. Clear? Now how we can do that? First of all, we can see that cell line A does not express human haptoglobin gene. Even the cell line D is also not expressing the human haptoglobin gene. So that means definitely the human haptoglobin gene is not present in cell line A and cell line D. Now the chromosomes present in cell line A is 1, 3 and 15. This is for cell line A. Similarly, from cell line D, we can see that 1, 2 and 15. These are the chromosomes which are showing positive in cell line D. But these chromosome does not contain the gene for haptoglobin. Why? Because in cell line A and D, the gene is not getting expressed. It is minus. So definitely the gene is not present in these chromosomes, because if the gene would have been in these chromosomes, it should get expressed. But it is absent from these two cell lines. That means the chromosome present in these cell lines definitely don't contain the gene for haptoglobin. So the gene is not present in chromosome number 1, chromosome number 2, chromosome number 3 and chromosome number 15. Now we left with chromosome 14, 16 and 21. Now here in 14 what we can see? The gene is not getting expressed. So definitely in 14 also the gene for haptoglobin is absent. In 21 also we can see that no expression of this particular gene. So definitely the gene is not present in chromosome number 21 also. So we left with only chromosome number 16, because in cell line B the gene is expressed, cell line C the gene is expressed plus, and here on chromosome number 16 also we can see that plus and plus is present for cell line B and C. So that means chromosome number 16 contain the gene for human haptoglobin. So this how we can map the presence of gene on any chromosome using the phenomenon of somatic cell hybridization. Now this question is for you. 5 cell lines are given. The presence and absence of this particular enzymes are given. You have to tell among these chromosomes on which chromosome the gene for this particular enzyme is present. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.